Hello, everyone. Welcome to our week 14 of our 15-week MCAT review course. So our week 14 lecture will be on Mendelian genetics, evolution, selection, and speciation. I will have 7 questions that we'll go over, and then we will go over the content related to those questions. Here on this slide, I have our GroupMe linked, our Instagram handle, our YouTube channel where you can find the other videos to our 15-week review course and all of the other videos that we have on our channel. And then we have one more week left. So next week we will have 2 lectures. We'll have a Monday recorded release at 8:30 and a Tuesday recorded release also at 8:30 as well. All right, so our first question here is going to be on Mendelian genetics. Again, when I go to the next question, pause, take a minute to answer the question, take a minute or two to answer the question, and then unpause the video and we'll go over it together and then go over the content related to that question. All right, and pause the video and we will go back in a second. So our first question here states, in the question below, brown eyes, capital B, is the dominant trait, and blue eyes, lowercase b, is the recessive trait. A man that is heterozygous for brown eye color is married to a woman who has blue eye color. What are the chances that a child will have the uppercase B and lowercase b genotype, so the heterozygous genotype, and also be a boy? All right, so when we are approaching this question, we first have to draw out a Punnett square. So we will draw out a square with 4 other squares inside. We will draw the alleles of each parent on each of the vertical and the horizontal aspects of the square. So that you can see here. We will go over the answer after I explain this. And then next we will draw we will fill in the squares with the zygotes genotypes that would result from combining each allele. So that is what you see here. And then now we will determine the phenotype of each genotype. So you can see here on the top row that we have 2 uppercase B's. So this will definitely be the, this is the dominant homozygous zygote. And here we have the heterozygous zygote. For the phenotype, we will both have brown eyes because uppercase B is the dominant. And then for our genotype, we have 50% homozygous dominant, and we have 50% heterozygous. So the genotype 50-50, the phenotype, all will have brown eyes. And then now we can find the probability of each genotype and phenotype. So we have a 50% chance then of having the uppercase B, lowercase b genotype, so the heterozygous genotype. And then because we are either getting a male or we're getting a female, that will also be 50%. And then we multiply those together in order to determine our answer, which is then 25%. So that is the answer to our question here. And so the answer is C. So now we'll go over a little bit about Mendelian genetics and the concept behind getting the answer from the Punnett Square. So most of us know about Gregor Mendel, who was the monk that studied inheritance patterns in pea plants. So what Mendelian genetics is, it's the statistical patterns of how traits are passed on from parents to offspring. So what Mendel did is that he noticed patterns of inheritance when mating tall and small and green and yellow pea plants. And then because of the advancements of science, we know the molecular basis now of heredity, but Mendelian genetics is the basis for this. So it's still important for us to understand more complicated scenarios of heredity and inheritance, especially ones that don't necessarily line up with dominant and recessive traits, more like codominance and incomplete dominance, which we will talk about a little bit later. And some important definitions that we have here One is what genes are. Genes are sections of chromosomes that carry information that determine traits. And what chromosomes are specifically is that this is what contains all of the individual's genetic material. We have 23 pairs of chromosomes. So from each parent, we get 23 chromosomes. Saying this instead of just saying 46, that we have 46 chromosomes, this reminds us that we have a maternal and a paternal copy. This is important because we can have different alleles for each gene, coding for something different, which makes us unique. So alleles specifically are alternative forms of genes. We can have homozygous chromosomes, which mean that each allele of the gene is the same on the maternal and paternal copy. For heterozygous, this means that alleles are different. So we have two different copies of the alleles on each maternal and paternal copy of the chromosome. And then if the allele is hemizygous, this means that we only have one allele and we don't have a dominant or recessive, it's just one. Some other important concepts is the law of segregation. This is two alleles of an individual are separated singly and passed on to offspring. We also have the law of independent assortment, and this states that alleles of one gene will separate independent of alleles from other genes. So once you eventually do end up going over meiosis, I think it is important to then determine which parts of meiosis, both the law of segregation and the law of independent assortment, happen in. So this is kind of a kind of meshing the two Mendelian genetics and then the more complex science of genetics that we know today. So for the law of segregation, we know this takes place in anaphase 1 of meiosis 1, and the law of independent assortment happens at metaphase 1 of meiosis 1. So again, I encourage you to go over that when you are going over meiosis. So now the Punnett squares again. So what these do is they predict the results of a cross between parents with a combination of different alleles for a trait. So like we did in the previous question, when we're drawing out a Punnett square, what we're going to do is first draw the square with 4 other boxes inside. We're going to draw out the alleles of each parent on the horizontal and vertical aspects of the square. Then we're going to fill in the square with the zygote genotypes that would result from combining each allele. And then we will determine the phenotype of each genotype. So for example, if we have our uppercase Y as dominant, and that is yellow, lowercase y is green, and that is recessive. So here, when determining the phenotype of each genotype, so we know that the phenotype here of the heterozygote is going to be yellow, and the phenotype here for the lowercase y, the homozygous recessive, is then going to be green. And now the probability of each genotype is going to be probability of the genotype heterozygote is going to be 50%. Probability of the recessive homozygote is going to be 50% as well. And the probability of each phenotype is also 50% for yellow, 50% for green. All right, so question 2, you can pause the video when I go over, we will go over the question. All right, so the question here states, if an individual has B positive blood type, he can donate blood to individuals with which genotype? So here I will first choose the Roman numeral to go over that appears exactly twice. And in this case, I can go over Roman numeral 2, or I can go over Roman numeral 3 first. Here I will just start and go over with Roman numeral 3. So blood type, we have A, B, and O blood type. These code for the antigens on red blood cells. So this individual that has B positive blood type, he is going to have the antigen on the red blood cell for the antigen B, and also the antigen for the rhesus factor. So if this person is donating blood to a person with B positive blood, then that person will also have the B and the rhesus factor antigen. So he can donate to individuals with that blood type. So therefore, I can cross out anything that doesn't have 3. So I can cross out A and I can cross out B. I know 1 is right, so I will evaluate 2. So if this individual who has B positive blood type donates to an individual with A positive blood type, this will cause a transfusion reaction. The body will reject this new blood because it doesn't have the B antigen, and it will create antibodies to destroy that these new red blood cells coming in. Just evaluating 1, AB positive. This is a universal acceptor because it has all the antigens as A, B, and positive. This is an important thing to understand for the MCAT, and one reason that we talked about it today is that this is an example of codominance. ABO blood type is an example of codominance, while the positive and negative part is actually classically dominant. Talking about the rhesus factor there. So therefore here, our answer is going to be C. 1 and 3 is correct. All right, so we will talk about complete dominance and incomplete dominance, or codominance and incomplete dominance as well. So what incomplete dominance is, this is when one allele does not completely suppress the other allele from being expressed. So an example of this is between white roses and red roses. When they mate, you can get a pink rose when you get the heterozygous version. This is because the red and the white aren't completely dominant and they don't completely suppress each other. Codominance, on the other hand, this is when alleles of a given gene are both expressed. So one isn't suppressed, but they're both going to be dominant and they're both going to be expressed. An example of this is blood type, like we had just discussed. We have allele A codes for antigen A. Allele B is codes for antigen B. And then the allele O, which can also be expressed as lowercase I, does not code for an antigen. And then again, as we discussed, the allele RH codes for the antigen RH factor, which is the rhesus factor. And these alleles are classically dominant. Again, this is an important thing to understand for the MCAT, so make sure that you are understanding this as codominance and being able to understand what type of blood can be donated and what type of blood can be accepted depending on the blood type that the individual is. All right, so just a few more definitions here about genetics that we've been talking about. So one is pleiotropism. So this is when one gene controls unrelated phenotype, a unrelated phenotype, or multiple phenotypes. So an example of this is PKU. So what this is a mutation on chromosome 12. This gene normally wants to code for phenylalanine, but this mutation will then code for tyrosine. And then many different phenotypes can come out of this mutation. We see sometimes eczema, we see sometimes mental retardation, and then some other ones can happen as well. Another definition that we have here is polygenism. These are traits that are controlled by more than one gene. An example of this is skin color, because more than one gene determines what our skin color is. Another definition here, penetrance. This is the likelihood that individuals with a given genotype will express an expected phenotype. Expressivity, it's important to differentiate expressivity and penetrance. So what expressivity is, it's when a phenotype is expressed, but to a different degree compared to individuals with the same genotype. Lastly, we have epistasis. This is where expression of alleles for one gene depends on the expression of another gene. So this could be curly hair not being expressed because the gene for baldness is also expressed. All right. So question 3 here, we'll talk now about Hardy-Weinberg genetics. All right, so again, pause the video. We'll go over it in a minute. All right, so the question here states, if the frequency of the dominant allele is 0.25 in a population that is in Hardy-Weinberg equilibrium, what is the frequency of the homozygous recessive genotype in this population? So first, the Hardy-Weinberg equations are going to be what we can use to solve this problem. So first we'll write down what our value for P is and our value for Q. So our value for P is 0.25, as P is going to be the frequency of the dominant allele in a population. So here we have P is 0.25, Q, we're not sure, but using these two equations, we can then find what the answer is. So as you can see here, we're just plugging in the values to the first equation to get our value of Q, which is 0.75. And this is going to be the frequency of our recessive allele. We are then trying to find the frequency of the homozygous recessive genotype, which is going to be Q squared. So therefore, plugging in the values that we have here, 0.25 and 0.75, we then get our answer is about 0.56. So that we can see as our answer is B. All right. So the Hardy-Weinberg principle, what this is that it states the frequency of alleles in a gene pool of a population will not change over time as long as the following conditions are true. So if these conditions are true, we are in what we call a Hardy-Weinberg equilibrium. So these conditions are that there's no mutation, there's no migration, there's no natural selection, there is random mating, and the population is sufficiently large to avoid random drift, which we will talk about later. And then so an important thing to understand also here is what a gene pool is. So the gene pool is the sum of all the genetic information from all individuals in the population. And it's also important to note that Hardy-Weinberg is based on population, not based on individuals in the population. And then again, the equations that we need to know are P plus Q equals 1, where P is the frequency of the dominant allele and Q is the frequency of the recessive allele. The other equation, P squared plus 2PQ plus Q squared equals 1, where P squared is the frequency of the dominant homozygous genotype, Q squared is the frequency of the recessive homozygous genotype, and 2PQ is the frequency of the heterozygous genotype. Therefore, if we know P and Q, we can determine our which is our allele frequencies. We can then calculate the frequency of the genotypes in the population or vice versa. All right, so question 4 here is also on Hardy-Weinberg genetics. So I'll give everyone a couple minutes to do the next question, pause the video, and we will go over it. All right, so the question here states, which of the following will not disrupt Hardy-Weinberg equilibrium? So we have A, genetic drift, B, mutation, C, random mating, or D, migration. All right, so we will talk about, so we talked about what makes Hardy-Weinberg equilibrium happen. So the one here that would be the outlier would be C, random mating, as we know that we need random mating in order to have Hardy-Weinberg equilibrium. We cannot have genetic drift, mutation, or migration. And we'll talk about here in the next slides that these are things that happen in real life, but when discussing Hardy-Weinberg equilibrium. These are just things that we take to be true. All right. So as I was saying, why Hardy-Weinberg equilibrium doesn't actually exist in reality. One reason is because of genetic drift or random drift, which is random events that can alter allele frequencies significantly in a small population. So if you look at the diagram here, genetic drift happens more likely in small populations. So let's say we started with 10 alleles here, all with, you can see if you have different colors would be different alleles. After a lot of generations, we now only have 6 of the original allele left. 6 of the original alleles are present here in this generation. And then after even more generations, we only have 2 left. And this is just because we have a very small population. So it is likely that only a few alleles then reproduce. And eventually you just get with smaller and smaller alleles left in your population. 2 examples of genetic drift. One is the bottleneck effect. So this is when a major disaster kills off a significant portion of the population. This alters allele frequencies. For the founder effect, this is when a portion of the population emigrates and creates a population with a different gene pool. For genetic drift, it's important that you know that traits that end up being in the population after many generations, these ones aren't selected for. It doesn't mean that they are more fit. This is just based on random chance. Other reasons here is that we have mutation. These are change in the coding sequence, as you can see here, on DNA. This is because we will always have inborn errors in DNA polymerase. We'll also have mutagens that can cause mutations. And we do have non-random mating, which is when individuals in the population preferentially select their who they mate with. All right, our 5th question here. Now we'll move on to evolution specifically. So I'll give everyone a couple minutes to answer this question as well. All right, so our 5th question here states, which of the following will have the greatest fitness? Roman numeral 1, a shark that has 3 offspring and nurture them to maturity. 2, a fish that has 9 offspring but abandoned them and only 3 of the offspring survive. Or 3, a squirrel that has 20 offspring but only 5 of the offspring survive. So here I will evaluate Roman numeral 2 as it appears in the answer choices exactly twice. So evaluating 2, I have a fish that has 9 offspring but abandoned them and only 3 of the offspring survive. So this is the same as 1, because when I am thinking about fitness, I'm just thinking about which is contributing most to the gene pool. So there's going to be 3 offspring in each, but when I'm looking at Roman numeral 3 here, there's going to be 5 more offspring, so more alleles in the gene pool. So therefore 3 is going to be the most fit. Therefore my answer here is C. All right. So now I'll talk about evolution, natural selection, and I will also talk about fitness and fecundity in the next slides. So what evolution is evolution is the change in heritable traits in a population over a period of time. It's important to note that this takes place in a population, not just within individuals, and this occurs by natural selection. It can also occur by genetic drift, but it mostly occurs by natural selection. So what natural selection is that one trait that is advantageous over another is selected to be passed on to more generations versus the other traits. And we call it natural because it's all based on who has the greater probability of surviving and reproducing, which is fitness, and we'll talk about that later. So when looking at our diagram over here, let's say we have 50% of these black beetles and 50% of these beige beetles. Let's say being a black beetle makes them more likely to survive and reproduce because they can blend in better. So over time, the allele frequencies will change and the black beetles will be more present in the population after different generations. It's also important to note that evolution only occurs to heritable traits and not acquired traits. So let's say getting strong throughout your life, that won't necessarily pass on to future generations. That will just stay there as an acquired trait. All right. So as I said, fitness is the ability to pass on traits to their offspring, meaning to add more alleles to the gene pool. And to put simply, it's the ability to survive and reproduce. When looking here at this diagram, we can see that this brown beetle here versus green beetle has a higher fitness because it reproduces more and adds more to the gene pool. And so they have more beetles surviving that have a certain type of, or that have certain alleles. Fecundity, on the other hand, is the ability to reproduce offspring. And fecundity is selected for by natural selection. So let's say you have a population that has two different traits. Let's say we have one that has blonde fur, one has brown fur. And they might have the same chance of survival, but the brown fur individuals are going to have a higher fecundity if they just produce more offspring. So over many generations, you will have more individuals with this brown fur because there will just be an increased amount in the population, and therefore the allele frequency is going to be changed and tend more towards the individuals with brown fur. All right, so question 6. We'll talk about different modes of selection, so not necessarily just natural selection that we were talking about. All right, so pause the video and get the answer to this question and unpause it when you are ready. All right. So our question here, consider a species of beetles that migrate to a region where it is advantageous to be larger, and this extreme phenotype is then selected for in subsequent generations. What is this mode of selection called? All right. So evaluating each answer choice here, I can determine that my answer is going to be C, directional selection. We'll go over the different modes of selection here, but the reason is because directional selection is selecting for an extreme phenotype, one on each side of the spectrum and not just in the middle, because it selects for the large beetle. This is going to be directional selection. All right, so discussing each of the different modes of selection, we have firstly directional selection, which is a mode of natural selection where the extreme phenotype is favored, which we discussed. Another example, and not the one that it was in the previous question, is giraffes. So tall giraffes are selected for because they can get food easier, while the small giraffes are selected against because they have a harder time getting food off of getting their food, which are leaves, off of taller trees. So as you can see here, it's going to be, you're going to have more tall giraffes in the population. Stabilizing selection, this is a mode of natural selection in which both extremes of a phenotype is selected against, driving the population closer to the average. So an example here is of birds being an average size. It is easier for them to mate when they're not too large and they're not too small. So therefore, there's going to be more birds that are of kind of the average size. Another mode of selection is divergent selection. So this is a mode of natural selection in which both extremes of a phenotype is selected for. So as you can see here, you're getting both extremes. So different than directional selection, because that's just to one side. The divergent is to both sides. An example of this is deer. If deer are too large, they will be able to fight, but if they are smaller, they can hide, but in the middle, they're not necessarily great at either. All right, so a few more. We have 4 here is kin selection. This is a mode of selection that does not benefit the individual, but others that they might live with socially. So an example of this is altruism. Let's say we have a animal like a squirrel that encounters a bear with its offspring. And if she chooses to save her offspring and sacrifice her own life, this is beneficial in terms of reproduction because her offspring can then pass on their alleles and add more to the gene pool, while she, let's say if she is past reproductive age, she wouldn't be able to add more to the gene pool, so it benefits their species as a whole. We also have sexual selection. This is a mode of selection in which animals do not choose their mates randomly, but by mating calls, rituals, and attractiveness. And lastly, we have artificial selection. This is selection in which humans intervene to mate certain plants or animals in order to get a desired trait. An example is the domestication of dogs. All right. The last question here, question 7. This is going to be about speciation. All right, so go over the question, take about a minute, and then unpause when you are ready. All right. So the question here states, when a horse mates with a donkey, a mule is born. What form of reproductive isolation is this? All right. So going over A, ecological isolation, B, gametic isolation, C, sexual isolation, and D, hybrid sterility. So a mule is born, which is a hybrid. And so this might be a post-zygotic barrier. Both ecological isolation and gametic isolation are pre-zygotic barriers. And C, sexual isolation is not a thing. So I know A, B, C is all wrong, while D is the correct answer, hybrid sterility. We will talk about all of the barriers here in a second. All right. So what a species is this a group of organisms that can reproduce with each other sexually? The species definition is different for organisms that reproduce asexually, but this is the definition for organisms that reproduce sexually. So what reproductive isolation is, these are the barriers that stop organisms of different species from having offspring with each other. We know that species are only different species because they cannot reproduce with each other. So what the pre-zygotic barriers are ecological barriers. So this means they can't mate because they exist in different environments. Temporal barriers. This is the barriers that make organisms unable to mate because they mate at different times of the year, day, or season. Mechanical barriers are the barriers that do not allow organisms to mate because the reproductive organs are incompatible, and gametic barriers is when organisms cannot mate because the sperm and egg of organisms are incompatible. So now post-zygotic barriers, first we have hybrid viability. These are when organisms can mate, but the offspring dies as an embryo. Hybrid sterility, organisms can mate, they can produce offspring, but the offspring itself can't breed. And then hybrid breakdown, organisms can mate, create hybrid offspring that can also reproduce with other hybrids, but for some reason, the second generation is unable to breed. All right, so now speciation and types of evolution. So speciation, this is the creation of a new species. I'll talk about the different types of evolution, and then two ways that you can create different types of species. We have divergent evolution and parallel evolution. So first here, what divergent evolution is. This is when groups of a certain species has different selective pressures causing speciation. Convergent evolution, this is the opposite. So this is when two species have the same selective pressures, causing them to gain many of the same characteristics. So this is kind of like dolphins and sharks. Parallel evolution is when two species go through a very similar evolutionary process due to similar selective pressures. And then coevolution, this is when two different species actually influence each other's evolution. I also want to mention what selective pressures are. So these can be environmental, like weather and location. It can also be biological, like diseases. And then they can also be related to resource availability, like having enough food and having enough water. All right, and that is it for week 14 lecture. We have 2 more lectures next week. So tune in. Again, if you have any questions, feel free to email me or ask questions in the group me. All right, good luck studying, everyone. See you next week.